Hello friends, welcome back to my channel. Hope you all are well and doing good. In my previous video, I talked about restriction endonucleases as a tool for recombinant DNA technology और आज मैं बात करूंगी cloning vectors की जो कि second tool है recombinant DNA technology में. So what I am going to cover in cloning vector is what are cloning vectors, their types, an understanding on the terms like copy number and transformation, and features of the cloning vector, in which I am going to cover origin of replication and selectable marker. The rest properties of cloning vectors will be covered in my upcoming videos. So stay with me and let's start our topic. All of us are aware of the process of cloning. In cloning, our gene of interest, or you can say foreign DNA, is inserted into a vector DNA with the help of the enzymes like restriction endonucleases or ligases, which will become a recombinant DNA, and this recombinant DNA is then inserted into a suitable host where it will get multiply or express itself to form a desired product. Now, here's the question arises, what are the vector DNA or cloning vectors? See, cloning vectors are the DNA molecules that carry a foreign DNA segment into the suitable host. So, let's have a look on the definition of cloning vectors. A cloning vector is a small piece of DNA that can be stably maintained in an organism and into which a foreign DNA fragment can be inserted for cloning purposes. Now, what are the different types of cloning vectors that can be used in recombinant DNA technology? Recombinant DNA technology में हम कौन-कौन से vectors use कर सकते हैं? See, the cloning vectors that are used in recombinant DNA technology may be your DNA that is taken from viruses or cell from higher organisms and plasmids from a bacterium. Generally, they are categorized into three types, that are plasmid, bacteriophages and cosmids. Let's have an understanding on all these vectors one by one. Most of the people are aware of the term plasmids. Plasmids are the small circular DNA molecules that are present in the bacterial cells. They are separated from the bacterial chromosomes and they have ability to replicate independently of it. Now, let's have a look on the statements of plasmids. Plasmid is a small, often circular DNA molecule found in bacteria. They are separated from bacterial chromosome and replicate independently of it. They generally carry genes that are associated with antibiotic resistance. Now, the second cloning vector that is used in recombinant DNA technology are your bacteriophages. As the name suggests, they are the viruses that infect the bacteria. Now, the question arises, why we used plasmid and bacteriophages as a cloning vector in recombinant DNA technology? हम क्लोनिंग वेक्टर्स के तौर पे प्लाज्मिड्स और बैक्टीरियोफाजेस को ही क्यों लेते हैं? द रीज़न इज़ दैट बिकॉज both have ability to replicate within the bacterial cells independently of the chromosomal DNA. The third cloning vector that is used for recombinant DNA technology is cosmid. As the name suggests, they are the hybrids of plasmids and lambda vectors. These are the plasmids that contain the cos sites of lambda phage. Now, let's have a look on the statement or content of the cosmids. Cosmids are the hybrids of plasmid and lambda phages. The plasmids in which the DNA sequence of cos sites of lambda phage is present. Cosmids have higher cloning capacity, say twice that of lambda vector. Now, what the terms copy number and transformation stood for? Copy number is defined as the number of vectors present in a cell, or higher the copy number, higher is the possibility of the vector to be present in a genome. For example, if you linked an alien piece of DNA or foreigner DNA with the plasmid or bacteriophages, then its number can be multiplied equal to that of the copy number of bacteriophages or plasmid. Now, the next term, transformation, it is the process of transferring the gene of interest, or you can say the foreigner DNA, into the host bacterium. Now, let's have a look on this term. Copy number is defined as the number of copies present in a cell. Transformation, it is the process through which a piece of DNA can be introduced into a host bacterium. Let's have one understanding on the properties of cloning vector, or what are the features that make a vector to be a good clone vector. The first one is your origin, or you can say the origin of replication. All of us are aware of this term, that this is the sequence that is used for the replication or to start a replication within the host organism. Now, let's have a look on the statement of this origin. It is a sequence from where the replication starts. Any piece of the DNA when linked to this sequence can be made to replicate within the host cell. Now, please pay attention to the statement. This sequence is responsible for controlling the copy number of the linked genes. If we want to get a higher copy number of the linked gene or the target sequence, we have to choose the vector whose origin sports a higher number. The second property of cloning vector is the presence of selectable marker. All of us are aware of the procedure of cloning. Here you can see in this diagram, this is your recombinant DNA which is inserted into the host organism. Now, whether the host organism get transformed or not is very difficult to tell. When we grow the host organisms on the Petri plate, we get a number of bacterial colonies, and to screen whether the host organism get transformed or not is a very tough job. Here comes the role of selectable marker. कौन सी जब हम लोगों ने पेट्री प्लेट के ऊपर बैक्टीरियल कॉलोनी ग्रो करी, तो ये जानना कि कौन सी कॉलोनी ट्रांसफॉर्म हुई है या नहीं हुई है, एक बहुत ही टफ जॉब है. और यहाँ पे रोल आता है सेलेक्टेबल मार्कर्स का. Selectable markers are the sequence of genes whose expression allow us to identify the vector is introduced into the host cell or not. Selectable markers वो sequence होते हैं जीन के जिनकी प्रेजेंस की वजह से हमें ये पता चलेगा कि जो हमारा होस्ट ऑर्गेनिज्म है वो ट्रांसफॉर्म्ड हुआ है या नहीं हुआ है. Selectable marker are of two types, positive selectable marker and negative selectable marker. Positive selectable marker are those which allow the host bacteriums to grow, and most of the antibiotic resistant genes come under this category. For example, E. coli cells do not carry any resistance against antibiotic, but if they get transformed, then they code for the genes that encode for antibiotic resistance, like amplicin, chloramphenicol, kanamycin. Now, let's have a look on this. Positive selectable marker are those whose presence allow the host organism to grow. For example, antibiotic resistance gene. Genes encoding resistance to antibiotics such as amplicin, chloramphenicol, tetracycline, etc. are useful selectable marker. Negative selectable marker are those whose presence will not allow the host organism to grow. Now, with our discussion on selectable markers, we can conclude the function of selectable marker is it helps in identifying and eliminating non-transformants and selectively permitting the growth of transformants. With this, I am ending my video now, and in my upcoming video, I am going to cover the rest of the features of cloning vector and competent host. Please stay with me, watch my videos, like them, share them with your peer group, and please subscribe to my channel. Till then, bye bye.